again, it's going to just be a brief overview of the chapter. It's not going to cover the entire thing. I think also inside this week's online portion, you'll have some of this as well. First of all, what is organic compounds? Do you guys remember that? Yes? No? Remember? What kind of atoms or elements are in organic compounds? Carbon and hydrogen. Very good. I read your paper. Cheater. They're also called hydrocarbons when we're talking about hydrogen and carbons. The thing about organic compounds is that they have a functional group. And in this particular case, this diagram here just shows it as a side group, but a functional group usually has some kind of chemical reaction that gets involved with a chemical reaction, so that that's why it's called functional group. The biomolecules, these are biological molecules that make up life. So they are found within living organisms, such as carbohydrates, proteins, nucleic acids, and lipids. Very good. So yeah, all these are organic compounds here. They're all composed of carbon and hydrogens. So all these are... Let's see. Is carbon dioxide an organic compound? CO2? No. Why is it not? Why is CO2 not an organic compound? Yeah, there's no hydrogen, right? So it's not an organic compound. So if it's not an organic compound, what is it? No. Inorganic compound. Very good. Very good. Okay, macromolecules. These are large molecules. Hence the name macro, right? Large molecules. So we do find this in carbohydrates, protein, and nucleic acid. Out of the four biomolecules, which one's missing here? Lipids, yeah. So lipids, we don't really have a macromolecule for it, but so polymers. Polymers form these macromolecules. Polymers, poly meaning many, yeah, So there's many pieces to this. Over here, you look at it, this is all carbohydrates. A polymer of carbohydrates are all shown here. We have a polymer of a protein, and here is a polymer of a nucleic acid. Monomers, on the other hand, is the simplest form of that particular macromolecule. So if we're talking about carbohydrates, what is this? Can you guys remember? You guys remember from last unit? I kind of showed this. Yeah, it was glucose, right? Yeah, So it was glucose, very good. Here is an amino acid. So an amino acid is just one of these within this polymer, while the glucose is just one of these in this polymer here. Over here is a nucleotide, and it's going to consist of one part of this nucleic acid. So these are the simplest form monomers. And what would you expect if there's two monomers that form a molecule, what will it be called? What's a prefix of two? Okay. Dimer. It would be a dimer. How about if you had three of them? Trimer. Yeah, very good. Boom. So how do we form these polymers? Is doing this dehydration synthesis. It's called dehydration synthesis or condensation due to this polymer's two shorter molecules. When they form a bigger molecules, they are going to release a water molecule. And so that's why it's called dehydration. A water molecule is taken out of the taken out from this reaction here. So two shorter polymers making a bigger polymer is going to undergo the dehydration reaction. So when we're talking about a monomer and a monomer forming a dimer, it's going to undergo this reaction here, the dehydration reaction. Yeah. And then hydrolysis, like the name implies, we're going to use water, hydro, right, to cut something, lysis, to cut something. So here's a larger molecule of a larger polymer broken down into smaller polymers due to having water being involved in this reaction. Okay, it's just the reverse of the dehydration reaction. If you look at it, if you just flip this upside down, it's going to be the dehydration reaction. That makes sense? Yeah? So carbohydrates are our first biomolecule we're going to talk about. There's this equation here, CnH2nOn. I do want you guys to kind of know this. It's just a formula of all kinds of carbohydrates. So the simplest form of carbohydrate, we mentioned one earlier. It was what again? Glucose, yes, glucose has C6H12O6. Okay, so this N is just showing you how many of these atoms are within that molecule, and the simplest one is 6. So it'll be C6H12O6 is the simplest form of a carbohydrate. So what would if you have two glucose formed together, it's going to be called maltose. What's going to be the formula for that then? So if there's two glucoses forming a dimer, or in this case it's going to be called maltose, what is the chemical formula for that then? C12H24O12. Very good. Okay, so carbohydrates, we use it for energy for the most part, and also we use it to store energy as well within animals and plants. And if we look at here, the monosaccharides, this is the simplest form. This is the monomers that we're talking about for this biomolecule, are the monosaccharides. And the monosaccharides are going to consist of glucose, galactose, and fructose.
Is it the same formula for glucose?
Yes, all of them are the same formula for glucose. However, as you can tell, they look differently, right? So if we were to count, if you look at this, they all look different. Here we have, it's very similar to glucose and galactose are very similar, but you notice how there's this hydroxyl group, this OH and H. It's flipped around here for the galactose. And for fructose, it's clearly different because now we have a pentose versus a hexose over here. But however, we still have that 6 carbon down here. Okay, so it's still 6 carbon molecule here. It's just formed differently. And fructose tends to be the sweeter of all the sugars. We'll talk more about that. A disaccharide. So under what reaction will we form a disaccharide if we have two monosaccharides together? We just said it in the last slide. Dehydration, yeah, synthesis or condensation. Okay, so here we have disaccharide. We have a dimer of glucose or sugar molecules due to that dehydration reaction. And the ones I will need to know is sucrose. Sucrose is a combination of both fructose and glucose together. And this is our common table salt that we use on... Yeah, table sugar. Yeah, not table salt, table sugar. Lactose, on the other hand, is galactose with glucose together to make milk. Okay, so that's the milk sugar that we're talking about. Uh-huh. It does have fructose. I mean, sorry, table sugar, not table salt. Yeah, I got that confused. Yeah, it does have fructose in it. It's a glucose and fructose together, forming sucrose. Even raw sugar is, I think, the same thing too. It's not pure glucose. When you have actually pure glucose, like glucose and glucose together, you form maltose, okay? So maltose is found in starch. So if you break down starch into smaller starch molecule, if you have a dimer of it, it's going to be maltose, okay? Glucose and glucose together. Yeah, in beer as well. Yeah, it'll be maltose. Yeah. Polysaccharides, like the name implies, this is when we have it as a polymer, right? It has multiple chain of monomers together. So this is our complex carbohydrates. We have glycogen, and this is how animals and how we store our glucose. So when we have an abundance of glucose, we're going to store it. We're going to store it in our liver and also in our muscles as well. That way, whenever we need to utilize the glucose for energy, we just cleave off one of these molecules and then utilize the glucose for our cellular respiration, which we'll talk about next time. Starch is how plants store up their sugars. And as you can notice, the difference between glycogen and also starch is that the bonding between the glucose to glucose molecules is slightly different. Okay, notice how this bonds together with the glucose and just forms this polymer here, while this one branches off into different branches. So this is found in bread and rice and other kind of potatoes. So anything that has starch in it will have this particular polymer here, polysaccharide. And cellulose here, this is it forms the cell walls of plants, and that's why it tends to be a lot more sturdier in what makes the bark of a tree. And this cellulose is another way of forming the glucose and glucose link together. We cannot digest cellulose, and that's why it becomes a form of fiber for us to be able to help regulate us in passing our feces. Chitin. This is what's on the insects and crustacean. It makes the shells of it. And it has yet another linkage between the glucose molecules. Okay. And this last picture, I wanted to just show you that although we do end up having starch within the corn that we harvest, what happens is that after the starch, we know that starch is made up of what molecules? What monomers? Glucose. Yes, it's keep on a long chain of glucose. Okay. However, because we end up wanting to manufacture these products to become sweeter and more appealing to people, we convert that into fructose. And fructose, like I said earlier, is a sweeter sugar than glucose is. And that's why we have those high fructose corn syrup in a lot of our products to help us consume more of these. Unfortunately, if we end up consuming more of these, it's going to cause problems with the amount of calorie intake that we have, cavities, and so on and so forth. Okay, lipids. These are the biomolecules that are actually hydrophobic. And what does hydrophobic mean again? They're afraid of water. They repel water. So the first lipids, there's a different type of lipids, but the first one that we're going to talk about are the triglyceride. The triglyceride is the normal like fat that we talk about, and this is actually how we store our own energy. So if we have too much of it, not only do we store it as glycogen, this is when we have even more amounts of it than we can store as glycogen. So we end up storing it as fat. It helps us insulate our organs and our body temperature too. And so it consists of a glycerol, which is the alcohol here, as shown over here on this left-hand side of this molecule. And it contains 3 fatty acids, hence the name the tri part. Okay, the glycerol is the glyceride portion of it. So saturated fatty acids, this is when, if you look at this, the hydrogens take up all the carbon's bondings. So for each carbon bond, it will have 2 other hydrogen and then another bond to another carbon. So that's why it's saturated with hydrogen bonds. And so it has the maximum amount of hydrogen for each carbon. And that's why it tends to be more solidified at room temperature. So if you think about, can you guys think of other saturated fats besides butter? Lard. Anything else that's solid at room temperature? More or less? No, that would be more liquid, right? Cheese, yeah, very good. Unsaturated fatty acids, this is when we actually start to have, notice there's a double bond here. So it's not saturated now because this carbon only has 1 hydrogen bound to it, and then it has 2 bonds bound to this other carbon. So, and notice how it has a kink to it as well due to that double bond. And because if it's kinked, then they can't stack up with each other that neatly. And if you can't stack up each other that neatly, that's why it's a little more fluid-like. And so oils are unsaturated fatty acids. So the more unsaturated fatty acids, the more healthier type, because it tends to prevent plaque buildup within your arteries and blood vessels. And we do need fats in our diets too to help, as we will later learn, we will end up needing it for our plasma membranes of our cells, and also, once again, to store energy as well. It's just that nowadays we consume it a lot more than ancient times when we were hunting our food, right? Like, it would be very rare for us to actually kill an animal to eat it. Over here, we just purchased it at the nearest fast food restaurant or something like that. So, phospholipids. Okay, so talking about how it is necessary to have some certain fats to build up phospholipids. And phospholipids are found within our plasma membrane, which I'll talk about in the next, or at the end of this lecture. And they end up having, building up our cellular membrane around our cells. And then, as you can see here, it has a hydrophilic polar head and 2 hydrophobic fatty acid tails. And just looking at this, what kind of fatty acid tail is this versus this one here? The one on the right is, this one here is the saturated one, right? It's straight, right? While this one has kinks in it, right? So these must be double bonds over here. So this is a unsaturated fatty acid. Very good. Okay, finally, there's steroids I want you guys to know. Steroids are 4-ring structures, and and as you can see here, these are the 4 rings that we're talking about. These are found in cell membranes, although, yeah, I don't know. No, you don't see it. Yeah, maybe you do see it in that picture. Let me see. Yeah, you do see it. It's these little yellow these things here. If you look at them closely, there's like 4 ring structures there. So you do find them in plasma membranes, but it also helps us produce steroid hormones. So cholesterol is the parent steroid hormone that will end up producing the different types of hormones in our bodies. So males tend to have more testosterone than females, while females tend to have more estrogen and progesterone. However, there is still some testosterone that's produced within females and some estrogen within males as well. So it's just relative amounts. Okay, and then there are like THG. This is like the anabolic steroids where people end up injecting themselves. So as you can look at it, it's very similar in structure as testosterone, and so testosterone helps increase the amount of like muscle cells and bone density growth and stuff like that, and so it ends up increasing the amount of muscle fibers that they will be able to have. Okay. So some things I want you guys to look at this is be able to observe the different type of chemical structure. I don't want you to memorize them, but I want you to actually kind of identify them by how they look, kind of thing. You don't have to say, okay, this is the carbon that has to be over here and the oxygen over there. Not like that, but general shapes and structures I want you guys to kind of know. So for a protein, this is a polymer of amino acids, so a string of amino acids joined together. And they're joined together through a peptide bond. So if you have 2 amino acids forming or many amino acids forming a protein or polymer, what kind of reaction is that again? If you're forming smaller dehydration reaction, very good. So there's about 20 types of amino acids. The reason why I said plus is because there's actually a couple more amino acids, but these are the 20 that are naturally occurring within nature. That you don't have to memorize this, but some things I want to kind of show you is that, well, I'll show you in the next slide, but we have a functional group for each one of these, and as you can see, these are all the functional groups of them. But they all have in common, all the amino acids have in common is this structure here. The amino acid is the monomer. We have an amino group, as shown over here. It's the one with the nitrogen. Okay, and then we have a carboxyl group, which is shown in the pink side here. So this is the C double bonded with O and C with the hydroxyl group. And it's always bound with a hydrogen as well. Oops. I don't know what's happening. Oh, hydrogen and a functional group. So the functional group is a side group here, and as we can see, all these are the functional groups here, so on and so forth. But all of them has, what is this group called? For those of you in the back, this is the H2N over here. Yeah, amino group. While this COOH over here is the carboxyl group. Yeah, very good. Carboxyl. To form a protein, you will first have amino acids forming the dehydration reaction and with each other. So here's an amino acid forming, what kind of bond is this? when you have a bond forming with another amino acid forming with another amino acid, they form a peptide bond. And it's usually a covalent bond. It's usually a covalent bonding with each other. And so arginine and leucine, whatever. So anyways, what I'm trying to get at is that as you have these peptide bonds forming all together, this sequence of amino acids is called the primary structure. And it becomes a little more complex when you go into the secondary structure. This is when we actually start to have more bonding in between different regions of the primary structure. So it's kind of like as if this. And then we go to the other slide here. So it's like, okay, so this was a covalent bond, right, between these two amino acids. But if we're talking about this leucine forming some sort of interaction with this trypsin over here, now we're forming a different type of bond. It could be a hydrogen bond. It could be another covalent bond. It could be possibly an ionic bond, so on and so forth. And when we start to form those kind of structure, this one is in particular are hydrogen bonds between different parts of the amino acid sequences. So here we have the beta pleated sheet, and here's the alpha helix. Okay, so the alpha helix will have hydrogen bonds on different areas of the primary sequence. Beta pleated sheet also hydrogen bonding on different parts of the sequence as well. And when we're starting to have more interactions, so van der Waals interactions between different parts of the primary sequences, now we're going into the tertiary structure. Okay? And when we have multiple tertiary structures now together, so over here we have like 4 tertiary structures now kind of interacting with each other. This is a quaternary structure. And now we have all kinds of interactions. We have ionic bonds, we have the hydrophilic to hydrophobic interactions, all kinds of different bonds now together. And now we have the functional protein. Well, some of them are functional in the tertiary, but most of them are going to be at this structure complexity over here. Okay, so what we have here is actually hemoglobin. This is actually found in our red blood cells, and it's where it binds oxygen to our red blood cells to carry oxygen in our blood. If we end up heating up proteins at certain temperatures or certain pHs that's not normal, it's going to cause denaturation. Remember, what are the bonds that are kind of weak? The van der Waals and... Yeah, van der Waals is the weakest one, right? And hydrogen bonds will be the next weakest. So those are the ones that are going to be broken up if we end up heating it too hot or causing a pH that ends up taking hydrogens away or putting hydrogens into the equation here. And then we're going to cause it to denature. And once it denatures, sometimes it could form back to its normal self and be functional again. But something like an egg, if you end up heating it up in a frying pan, it's going to be denatured and it won't ever form back to its normal shape and form anymore. Okay, this is kind of a rehash. The amino acid is once again a monomer. There's about 20 types of it, at least 20 types of it. A dipeptide is when we have 2 amino acids forming a peptide bond through what reaction? Dehydration reaction. Very good. So water is released, right? Released from this reaction. As you can see over here, this is actually showing you here. We have the OH group here of the, what group was this? carboxyl group and this hydrogen here within the amino group, releasing hydrogen, so dehydration reaction, forming a what kind of bond? Peptide bond. Very good. Okay, so this is a dipeptide because we only have 2 of them. Tripeptide, of course, is 3 of them. Polypeptide is when we have more than 10 or 15 amino acids linked together, and this is where we have the polymer. There's many different functions of protein. I'll just go through a few of them. One is through communication. We'll learn more about this when we're talking about the nervous system, is that there are some receptors on the cell plasma membrane, and this is a neuron here releasing neurotransmitters or signals that will end up binding to these protein receptors here. These red little dots are the protein receptors. And once it binds to that, it's going to cause something to happen on this side of the cell. It will either inhibit it from responding or cause it to be activated. And so this is the communication portion of it. So there is membrane transport function as well. So these are the channel proteins here, as you can see. And these help carry things in and out of the plasma membrane, so in and out of the cell. So it will cause things to enter or exit type of thing. It can also be a catalyst. Catalyst is usually the enzymes, so protein enzymes, that will end up helping reactions to occur. And we'll talk more about this in the next lecture slide. Okay, recognition and also for protection. As you can see over here, we have some proteins called antigens on our red blood cells, and it helps us to recognize ourselves, to be able to go, okay, these are my cells, these are my cells. But if there's some kind of foreign material like a bacteria or virus or even someone else's blood potentially, then we're going to go, okay, this is a foreign thing. It's not us, so it might be dangerous to us. Let's attack it and get rid of it. So we have these antigens or on our plasma membranes, these glycoproteins, for us to be able to recognize ourselves and to help us combat foreigners. Correct. The DNA within the cells will form these proteins that will be embedded on the plasma membrane of the cells. And your DNA will form different type of proteins on your cells compared to my DNA. There are sometimes, especially if we're talking about blood type, we might have similar antigens in that particular case if we're the same blood type. Lastly, that I want you guys to know is movement. So there are motor proteins. So here we have a motor protein at the end of a sperm here to cause it to be mobile. We also have motor proteins across our muscle fibers to be able to contract whenever we flex. And this is just another example of the communication portion of it. This is a receptor, and this is a protein receptor here. And so what happens is that when some kind of hormone, this is a FSH receptor. FSH is a follicle-stimulating hormone found in both males and females, but it's more pertinent in females when we talk about it in the reproductive system. And so FSH will bind to this receptor, causing a chain of sequences to occur. And you don't have to know most of what I'm going to say here, but just know that these proteins are used for communication as receptors in this case. So here's FSH. It binds to the FSH receptor. That's going to activate the G protein, causing the activation of this adenylyl cyclase over here. That's going to convert ATP, which you do know, into cyclic AMP, which you don't know yet. And then cyclic AMP is going to activate this protein called protein kinase A. It's going to cause a series of events that will end up producing estrogen and also producing more receptors as well. Okay. Don't worry about that. That was more of my thesis back in the day. So, yeah. What you wanted to take away from that slide was just that there's communication involved with the proteins through the receptor itself, from a different cell to this cell here. Okay, there are different properties of a protein as well. Notice how proteins are so complex. One thing I want you to know is the ligand. It's also called a substrate if we're talking about an enzyme. And these are molecules that ends up binding to the protein. Okay, and there's also an active site on the protein itself that ends up binding the ligand. So here, in this particular case, here's the protein. Over here is the active site. So it's going to be able to bind a ligand. And if a ligand is, I'll show you in the next slide here. So one of the properties of protein is that it's very specific. Okay, the protein has a specific shape to it, and that shape has an active site over here to be able to bind to a particular molecule. In this particular case, this is the ligand. This triangle shape is the ligand. And so if it fits snugly with each other, then it's going to interact with each other. Okay. However, something like this ligand is not going to fit because the active site is not going to fit it. All right. So it's very specific. That's what I'm trying to get at. Okay, so affinity is also another property of proteins. So proteins tend to have different affinities depending on the different ligands. So there are some ligands that are similarly shaped as this. If we ended up having a different ligand, just color it yellow instead. If we're saying that the blue one has more affinity, then which one is it going to be binding more to, the blue one or the yellow one? The blue one, right? Since it has a higher affinity to it. So it tends to bind to it a lot quicker, so there's more interaction between that protein and that ligand. Okay. Saturation. And this is very similar to that carrying capacity kind of graph that we saw, right? So here is the amount of number of protein and ligand interaction. So as you can see, there's a point in time where there's a maximum amount of protein and ligand interaction. So no matter how much ligands we end up putting into this system here, there will not be any more reaction between the two. What I'm trying to get at here is that we're only set with a set amount of proteins. Okay, so just say we have 50 proteins. Okay, if we have 50 proteins, when we put a little bit of ligands in here, just say 10, all 10 of them will be reacting with some protein, right? So that's why we have this steep curve. And if we keep on putting more ligands in, now when we put 20, it's going to continue to interact with those proteins. All the way until if we end up having 50 ligands with 50 proteins. Now all of them are occupied. If we put another 10 into it, so we have 60 ligands, but we only have 50 proteins. So at this point in time, those 10 extra ligands won't interact with any more proteins anymore. And so that's where we meet our maximum, our saturation point. Does that make sense? How do we increase this? Add more proteins. Very good. If we increase the saturation point by adding more proteins in it, and so if we end up adding more ligands, those proteins can interact with them. Very good. Okay, also another property of proteins is that there's this potential competition involved with the active site. So if we have something like this amino acid transporter here, both of these amino acids are similar in shape, and so they're going to be competing for this active site. If the yellow one tends to first binds to this active site, the green amino acid won't be able to bind to it, so it's not going to be able to react with that particular active site on that protein. However, if the yellow one is not there, then the green one can interact with it. So there's this competition for the active site if we have similar ligands competing for the same active site. Okay, let's take a little bit break since